So in Chapter 7, we're going to be discussing techniques of genetic analysis. In this chapter, we'll be talking about various types of blotting techniques, including their use in RFLP analysis, as well as genetic fingerprinting, and how those are used in various types of genetic testing, as well as paternity testing. And finally, in this chapter, we'll be discussing polymerase chain reaction, PCR, and how that's used. So the first thing we want to do on this figure is just discuss blotting techniques. This is a very important figure because it sets the basis for the other figures which come up later on in this chapter. So in the various types of blotting techniques, if you want to analyze DNA, RNA, or protein, now regarding DNA, as we talked about earlier in Chapter 1, DNA is a huge molecule. The chromosomes are very long and it cannot be analyzed directly. What must first happen is DNA first has to be treated with a restriction endonuclease, which generates DNA fragments, sometimes referred to as restriction DNA fragments, and these fragments are then analyzed after the DNA is cut up. Again, the reason is DNA is far too large to be analyzed directly. Alternatively, regarding RNA and proteins, RNA and proteins are sufficiently small. They are analyzed directly. RNA is never cut up. Proteins are not cut up with enzymes. They are analyzed directly. Earlier in immunology, you had talked about Western blots, dealing with how you analyze proteins in blood. In the Western blots, again, proteins are not cut up. They're sufficiently small and analyzed directly. Now, all DNA fragments, all RNA, as well as most proteins are negatively charged. DNA is negatively charged because of all the phosphate groups, same thing with RNA, and proteins for the most part are negatively charged, with the notable exception of histones, which are positively charged. So if these substances, either DNA fragments, RNA, or protein, are subjected to an electrical field, these substances, these DNA fragments, all RNA, as well as most proteins, will migrate to the positive terminus. So from here on, I'm going to be talking about DNA fragments. So if you take your chromosomes, add some sort of endonuclease, you generate hundreds, if not thousands, of DNA fragments. If they are applied to an electrical field, so if this is some sort of gel, they're put at the top of the gel, and at the top of the gel is a negative terminus, at the bottom of the gel is a positive terminus. All of these DNA fragments will migrate to the positive terminus, and they're going to separate from one another. And what do they separate on the basis of? They separate on the basis of size. The largest fragments do not migrate very far, and the smallest DNA fragments will migrate the quickest to the positive terminus because they go through the gel much quicker. So this would be the case for DNA fragments, same thing with RNA. They'll separate on a basis of size, as well as proteins separating on a basis of size. The smallest proteins migrate fastest to the positive terminus. So after you take DNA fragments, apply them to an electrical field, all these DNA fragments will migrate and separate on the basis of size. Now, at the end of the electrophoresis, if you take that gel and look at the gel, you can't see anything, and the reason is the DNA fragments, as well as RNA and most proteins, are colorless. And so you can't see anything. So what's done is to take that gel, so let's assume this is a gel where all the proteins have separated on the basis of size. Then you take a piece of paper, and then side by side, you take the gel and the paper side by side, and now electrophoresis in the direction of the paper. All of these DNA fragments now will appear on this piece of paper at the right position. Again, they are resolved on the basis of size. All the largest fragments will be at the top of the piece of paper. All the smallest fragments will be at the bottom of the piece of paper. So on this figure here, if you take the fragments from the gel and then transfer them to a piece of paper, generally it's some sort of nitrocellulose, you look at that piece of paper, you still can't see anything. Again, the reason is that DNA fragments are colorless. So now I'm going to set that aside for a minute and talk about a side issue which is related to an application of what we'll be talking about shortly. Now, at the bottom of this figure, again, I'm just going to talk about another issue here. Let's assume this horizontal line represents the beta globin gene. And again, this is a gene which is part of a very long chromosome. What chromosome number is not important here. But this is the beta globin gene, which is a part of a very long chromosome. Let's assume within the beta globin gene there are a variety of, there are several palindromes, and that's shown by the vertical lines. So here's a palindrome here, 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 and here. So if you take chromosomes and add a particular endonuclease, in this example I'll be talking about, let's assume we add the endonuclease EcoR1, and at the vertical lines, this represents palindromes for EcoR1. What's going to happen is this endonuclease will cleave the beta globin gene at this side here, 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 and here. Now, EcoR1 will also fragment the rest of the chromosome as well as all 46 chromosomes. But in the vicinity of the beta globin gene, notice how we generate one small fragment, one medium-sized fragment, and a very large fragment that was generated from the fragmentation of the beta globin gene. So if you take chromosomes and subject them to endonuclease digestion with EcoR1, all of these fragments from all 46 chromosomes then are separated on a basis of size. And 3 of these fragments are coming from beta globin, but you can't see them. These are then transferred to a piece of paper. Again, you can't see any of these fragments. So now the issue becomes, how do you detect these fragments from beta globin? Well, what's done is to make a probe, a DNA probe, that's complementary to the gene of interest. What's the gene of interest here? It's the beta globin gene. So we know what the sequence of the beta globin gene is, and we can make a probe that's complementary to the beta globin gene. And so I'm going to illustrate this, the probe as shown here by the red horizontal line. So the red horizontal line represents a probe that we make in a laboratory or commercially purchase that's complementary to the beta globin gene. Notice how this probe, this red horizontal line, will base pair with and hybridize with the small fragment from beta globin, the medium-sized fragment from beta globin, and the large fragment from beta globin. So now let's go back to this piece of paper. Here's this piece of paper where we have hundreds, if not thousands, of fragments from all your chromosomes, and 3 of those fragments are coming from beta globin. So now we add this probe to reveal the bands of interest, and this is going to be a technique called hybridization, where the probe will look for the fragments, in this case here, from beta globin. So now let's bring in the, so we take this piece of paper with all the DNA fragments and put it in some sort of tray or flask, and then add a solution which contains the radioactive probe. So let's bring in the probe. Here comes the probe. Now what's going to happen is this probe is going to look at all of those fragments from your chromosomes, looking to base pair, looking to hybridize. And this probe will only base pair with 3 fragments. It's going to base pair with the large fragment, the medium-sized fragment, and the small fragment. The probe will not base pair with any other fragment. Why not? Because it's not complementary to any other fragment. But here, the probe base paired with a small, medium, and large fragment. Again, DNA is colorless, including the probe is colorless. You can't see anything. So now what's done is after you add the probe, it's going to base pair with the DNA fragments from beta globin. Then what's done is to take this piece of paper, you wash away all the excess probe, then take this piece of paper, dry it, and add over top of it an X-ray film. And then anywhere where the radioactive probe base paired with the fragments from beta globin, because the probe is radioactive, the radioactivity then will strike the X-ray film. And then you come back the next day. Anywhere where the probe base paired with the beta globin fragments, what's going to happen is there's going to be bands now start to appear on the X-ray film. And in this example here, there will be 3 fragments from beta globin that will appear on the X-ray film. There's going to be a small, medium, and large fragment from beta globin. I want to emphasize, this does not mean there are 3 beta globin genes. This means there are 3 fragments coming from the beta globin gene. So this is the basis of blotting techniques, in this case here, looking at DNA fragments using a DNA probe. In this next figure here are the types of blots that are used in, for detecting DNA, RNA, or proteins. And these are the names shown here on the left of this table are very important. You need to know the difference between a southern blot, northern blot, western blots, and what a dot blot is. And so when someone says, or on USMLEs, if you see the word southern blots, what that's telling you is DNA is being analyzed after electrophoresis of DNA fragments using some sort of DNA probe. Southern blot means looking at DNA fragments on a gel. Whereas a northern blot means you're analyzing RNA. And RNA after electrophoresis, again, there's different sizes of RNA. After electrophoresis, the RNA will separate on the basis of size. And how is the RNA being analyzed? Again, it's using some sort of radioactive DNA probe. So DNA is complementary to the RNA of interest. As you know from immunology, you talked about western blots, where you analyze proteins. After electrophoresis, to separate the proteins on the basis of size. How is the protein of interest being detected? Is being detected by some sort of radioactive iodinated enzyme-linked antibody. So some antibody that's radioactive or enzyme-linked will be able to detect the protein of interest. Now, an up-and-coming probe, a blot that's used in your profession is the dot blot, and the dot blot is used to analyze DNA, RNA, or protein. But the big difference between a dot blot versus southern, northern, and western blots is right here. There is no electrophoresis that's required to analyze DNA, RNA, or a particular protein. Now, in medical genetics, you'll be talking about dot blots in much greater detail. So we will not do it here in Chapter 7. That'll be discussed in medical genetics. But what we want to discuss here is southern blots and northern blots. And even westerns you've talked about, and you will see that in the tape in immunology. But what we're going now is to talk about southern blots and northern blots. And on this figure here is the use of the southern blot for RFLP analysis. RFLP. Now, what does that mean? RFLP analysis means restriction fragment length polymorphisms. Simply put, what this means is if you take your 46 chromosomes and get rid of all those histones, you got negative DNA. If you take your 46 chromosomes and put them in a test tube and add some sort of endonuclease, you will fragment all of your 46 chromosomes, and you'll find out that your DNA fragments will be different in size compared to my DNA fragments, which are different in size compared to your neighbor's DNA fragments. So the issue now becomes, why are your DNA fragments different in size, restriction fragment length? Why is your fragments different in size compared to mine, compared to your neighbor? Now, there are two reasons why your fragments, your DNA fragments from your chromosomes are different than my size of my fragments compared to your neighbor. There are two reasons for this. And one reason is due to these polymorphisms in the DNA fragments. The sizes could be different due to some sort of mutation. Now, that's what I want to explain on this figure here, is how polymorphisms generated by mutations can be analyzed. So what we got on this figure here are portions of two homologous chromosomes, two matching chromosomes. So here on let's assume this is chromosome A, this top part of this figure is chromosome A, and the bottom part is chromosome B. So these are two homologous chromosomes, matching chromosomes. And this is just a portion of one chromosome, chromosome A, and this is the same portion of chromosome B. Now, in chromosome A, at that portion, let's assume there are 3 palindromes at site 1, 2, and 3. These are going to be 3 palindromes for this endonuclease EcoR1. So if you take chromosomes, chromosome A, add EcoR1, it's going to cleave at site 1, 2, and 3. Why? Because there's palindromes for EcoR1, and you'll get fragmentation of the chromosome A at site 1, 2, and 3. So what these vertical lines shown here in the magenta is cleavage of those palindromes. Now, specifically, EcoR1 cleaves between the G and the A. It's not important here. Just know that EcoR1 cleaves that palindrome at site 1, 2, and 3. Now, by cleaving those three palindromes, notice the size of the fragments as illustrated on this diagram. When EcoR1 cleaves between site 1 and 2, there'll be a fragment of 1.2 kb in size, and when EcoR1 cleaves at site 3, there'll be another fragment being generated at 0.9 kb in size. So 2 fragments coming from this portion of chromosome A. Now let's assume in chromosome A at site 2, there is this GAATTC. This is the palindrome for EcoR1. Let's assume in chromosome A, there's that third nucleotide here, the A. In chromosome B, let's assume there was a mutation of that A to a G. Notice how this is no longer a palindrome. Now you might say, well, if you have a mutation, doesn't that cause a disease? You got to remember that 97% or so of your chromosomes are junk DNA. You can have all the mutations you want as long as you don't have a mutation within a gene. If it happens within a gene, certainly it can give rise to a disease, or it could be lethal. But here, if you have a mutation in a spacer DNA, junk DNA, it has absolutely no deleterious effect. So let's assume now in chromosome B, there was a mutation where the A in chromosome A was converted to a G in chromosome B. So there's no longer a palindrome at site 2, but there still is a palindrome at site 1 and site 3. So when chromosome B is then exposed to EcoR1, EcoR1 will cleave at site 1 and also cleave at site 2, at site 3, not site 2. Now notice how this fragment size is much bigger than this small fragment here and this small fragment there coming from chromosome A. So now in your book you see a question coming into play here. I'll give you a chance to look at this question and to analyze these 2 blots. Give you about 10 seconds or so to look at this. So what these 2 figures at the bottom, these are southern blots. Again, what's a southern blot? It's DNA fragments being analyzed by some sort of DNA probe. And what's the DNA probe? It's going to be shown right here. This is going to be some sort of DNA probe that we make in the laboratory or commercially purchased, which is complementary to a fragment of interest. And so if we take chromosome A, add EcoR1, we generate lots of fragments. Now this probe is going to be complementary to this part of chromosome A, and it's going to base pair with this part of this fragment coming from chromosome A. And what's the size of this fragment right here? It's 1.2 kb in size. Now that same probe, when it's added to fragments coming from chromosome B, that probe will also base pair with this fragment coming from chromosome B. And what's the size of this large fragment coming from chromosome B? That fragment is 2.1 kb in size. Where did that number come from? It came from 1.2 plus the 0.9. This site 2 palindrome was mutated, so that's why this is where the 2.1 kb fragment is coming from. Site 2 never got cleaved because there was mutation in the palindrome. Okay, so now what's done is you take somebody's chromosomes, which have either chromosome A or B, and now the issue is, what does a southern blot look like after fragmentation with EcoR1 and then probing with this probe? And the answer to this, which one of these 2 southern blots is correct? It's the one shown here on the right. Now, why is the one on the right correct? So let's assume now someone has a genotype AA. They have 2 chromosome A's. If they have 2 chromosome A's, in that case there'll be 2 fragments of 1.2 kb in size being generated, and that's shown right here. There'll be 2 overlapping fragments, each one being 1.2 kb in size if the person has the AA genotype. Now let's assume someone has an AB genotype. What would the banding pattern look like? In that case someone has a chromosome A. They'll be generating the 1.2 kb fragment, which will be showing up right there. And the other chromosome is chromosome B. That probe will see a 2.1 kb fragment, and that's illustrated right here. So someone who has a genotype of AB will now have two bands, one at 1.2 and the other at 2.1 kb in size. And lastly, let's assume someone has a BB genotype. So this person has two matching chromosome Bs, homologous. And so what happens there is when fragmentation is being done by EcoR1, it cleaves at site 1 and 3. In that case there will be two overlapping bands at the position of 2.1 kb in size. So this is why the southern blot shown here on the right is the correct answer. Importantly, it's the probe that determines which DNA fragments are going to be visualized in the southern blot. Now what I would like to do here is carry this one step further and illustrating that this probe will see the 1.2 kb and as well as the 2.1 kb. This probe will not hybridize with a 0.9 kb because it's not complementary to this part of the chromosome A. What I would like to do now is carry this one step further. How can we get a southern blot which does look like the one shown here on the left? So what I'd like to do here is now erase most of this material. Let's get rid of it. And now illustrate how the southern blot shown on the left could be the answer. So again, here's chromosome A, which has palindromes at site 1, 2, and 3. If we add EcoR1, it cleaves at these vertical lines shown here in the magenta, generating two fragments, 1.2 kb and 0.9 kb. And whereas in chromosome B, it cleaves at site 1 and 3, generating a 2.1 kb fragment in size. Now, the issue is, notice if we have a different probe. In the previous figure, the probe recognized this part of the chromosomes, whereas now let's assume we have another probe which spans from here to here. Now, if someone has chromosome A generating these two fragments here, the 1.2 and 0.9, what will this probe base pair with? What would it hybridize with? Well, certainly it would hybridize with a 1.2 kb fragment as well as the 0.9 kb fragment because this probe is complementary to this portion as well as that portion of chromosome A. Now, what about chromosome B? That probe will also base pair with a 2.1 kb fragment because it's complementary to it. So now, let's assume someone has an AA genotype. What would the banding pattern now look like? So if someone has an AA genotype, notice what's going to happen is this person's going to have two homologous chromosome A's. In both cases, they'll be generating a 1.2 kb fragment, which is shown right here. These will be two overlapping fragments at 1.2 kb in size. The 0.9 kb fragment also will be visualized by the probe, and that will be two overlapping bands, again, at a position of 0.9 kb in size. Now, what happens if someone has an AB genotype? In that case there, notice all three of these fragments will be visualized and recognized or being hybridized by the probe. Here's the 2.1 kb fragment coming from chromosome B. Similarly, the 1.2 kb fragment coming from chromosome A will be seen. And lastly, the 0.9 kb fragment will also be seen by this probe, and it'll be visualized down here. So this is if a person has an AB genotype. Now lastly, what would happen if someone has a BB genotype? A BB genotype. In that case there, this person would have two chromosomes, each one generating the 2.1 kb fragment, and there it is right there, two overlapping bands at 2.1 kb in size. So again, I want to emphasize, notice how the probe determines which DNA fragments are seen. Now, what I would like to do again is carry it one step further and ask yet another question. Let's assume we have chromosome A, which has these palindromes at site 1, 2, and 3, and chromosome B has a fragment at site 1 and also being cleaved at site 3. So now, let's assume the probe that we make in the laboratory or commercially purchased, let's assume this probe now will see these parts of the chromosomes. The question I have for you is, what would the southern blot now look like? Notice how this probe will base pair with a 0.9 kb fragment. This probe will also, that's coming from chromosome A. This probe also will base pair with the 2.1 kb fragment coming from chromosome B. Issue now becomes, what would the southern blot now look like? So if someone has an AA genotype, what would happen there is AA genotype, there'll be two overlapping bands at 0.9 kb in size. Let's draw it there, the two overlapping bands at 0.9 kb in size, if someone has an AA genotype. If someone has an AB genotype, what would the banding pattern now look like? In that case there'll be a band appearing at 0.9 kb in size, that's coming from chromosome A, and there'll be another band appearing at 2.1 kb in size that comes from chromosome B. If someone has a BB genotype, there'll be two overlapping bands at 2.1 kb in size. The bottom line to all this exercise is notice how there are three different southern blot profiles depending on which probe is being used. Previously we had a probe right here that recognizes this part of the chromosomes. Earlier there's another probe recognized this part of the chromosomes, and now thirdly we have a probe that recognizes this part of the chromosomes. And again, three different southern blots are being visualized depending on which probe is being used. So what have we done so far? This is RFLP analysis and looking at polymorphisms generated by mutations. Now there's another way in which we can end up with polymorphisms of DNA fragments, and this is done by polymorphisms generated by an item called VNTRs. First of all, what does VNTR stand for? What VNTR stands for is variable number of tandem repeats. And another name for VNTRs is, you might see it as microsatellite repeats, or you might see it as mini satellite repeats depending on how big the VNTR is. So here we got two homologous chromosomes. Here's chromosome A, and now at the bottom part, let's assume we have another chromosome. Its matching chromosome is not chromosome A or B. This is going to be another matching chromosome. Let's assume this is chromosome C. Now in chromosome A, if it's exposed to EcoR1, EcoR1 will cleave the palindromes at site 1, 2, and 3. So again, these are three palindromes at site 1, 2, and 3, and the EcoR1 will cleave here, here, and here, generating a 1.2 kb fragment and a 0.9 kb fragment. Nothing new compared to what we talked about earlier. Now, if in chromosome C, let's assume now in chromosome A, remember it has a palindrome at site 2 of GAATTC. Now let's assume in chromosome C at site 2, that palindrome is still there. It's GAATTC. There was not a mutation at this point here. So it is a palindrome GAATTC. Now what would happen to chromosome C when it was exposed to EcoR1? Well, EcoR1 would cleave here, here, and here because the palindromes are there and generating two fragments in chromosome C, in this vicinity of the chromosome. But now, let's introduce this business of the VNTR. Now, what a VNTR is a sequence of DNA, a very small sequence of DNA. And let's insert, because you've got to remember what a chromosome is. It's simply a stretch of nucleotides. Let's assume between site 1 and site 2, there is a sequence of DNA, ACAT-CCC. Again, there are going to be other sequences of DNA between site 1 and site 2. But within site 1 and site 2, there's a sequence of DNA, ACAT-CCC. Now let's assume in chromosome C, that sequence of DNA is amplified 10 times in tandem. That's illustrated here. ACAT-CCC 10 times in a row. ACAT-CCC 10 times in a row in tandem. That's where the name comes from. Variable number of tandem repeats. This is a repeat that's now been expanded 10 additional times in a row. Now you might say, well, does not have effect on genes and things. As long as it's not, this VNTRs is not in a gene, it's not a problem. You can have VNTRs in spacer DNA and have absolutely no problem with life. So this is, let's assume now this is going on in some sort of spacer DNA. So now, if chromosome A and C is exposed to EcoR1, what's now going to be the size of the fragments? So what's going to happen is we're going to get a fragment from chromosome A that's 1.2 kb in size. But now the fragment coming from chromosome C is going to be a little bit longer in size. Why? Because of that extra stretch of nucleotides. Again, the VNTR has been expanded 10 additional times. This fragment coming from chromosome C will be larger in size. Now, if we have some probe that will recognize this fragment coming from chromosome A as well as chromosome C, what would the southern blot now look like? So here on the lower right is going to be a southern blot. So if someone has a genotype AA, what would the banding pattern now look like? AA genotype. Well, in that case there'll be two overlapping bands at 1.2 kb in size. What would happen if someone has an AC genotype? What would the banding pattern then look like? Well, if someone has an AC genotype, the fragment coming from chromosome A would be 1.2 kb in size. Nothing strange about that. But now this fragment coming from chromosome C is going to be slightly larger in size. It's going to be illustrated here, just slightly larger in size. Compared to the 1.2, it might be 1.3 kb in size, but slightly larger in size because of this VNTR between site 1 and site 2. Now, if someone has a genotype CC, what would the banding pattern now look like? There would be two overlapping bands at roughly the position 1.3 kb in size. That's if someone has the genotype CC. So again, to summarize, what you see here is polymorphisms generated by VNTRs, as well as polymorphisms generated by mutations that we talked about earlier. Now, this is all the basis for using RFLP analysis for genetic testing, for forensic medicine, and for other types of testing of DNA. Now, on this figure here is an application of the VNTR. What we talked about earlier is how the basis of the VNTR and now the application of the RFLP for diagnosing of sickle cell anemia. Now, in your book, what you have in your book is the lower part of this figure. What you see at the lower part of this figure is a pedigree for sickle cell anemia. Now, you know from medical genetics that sickle cell anemia is an autosomal recessive disease. So in this pedigree, here are two parents. Here is a male and a female, and giving rise to the offspring. Here is a male who has sickle cell anemia. Here's a daughter who does not have the disease, and here is a fetus. Now, what they might do on a national board is they might show you a pedigree like this and have an arrow pointing to the fetus and with a big question mark there, and say, they might ask you, what is the outcome of this fetus? What is the probability that the fetus is homozygous normal, homozygous with the disease, sickle cell anemia, or heterozygous for sickle cell anemia, a carrier for the sickle beta globin allele? So based on the pedigree alone, what is the probability that the fetus is a carrier for sickle cell anemia? And the answer is, based on the pedigree alone, the fetus has a 50% probability of being a carrier for sickle cell anemia. Why? Because this is sickle cell anemia is an autosomal recessive inheritance mode of transmission. So what this means is the father as well as the mother must be carriers, and they're passing on the trait to the fetus with a 50% probability of being a carrier, a 25% probability of being normal, and a 25% probability of being homozygous for the sickle beta globin allele. So now that's based on the pedigree alone. All you can say is probability. But now with the use of the power of the RFLP analysis, we could determine precisely what is the outcome of the fetus. Now at the top of this, before we talk about the RFLP analysis, which is shown at the bottom of the figure, and it's also in your book, what I'd like to do is talk about the top of the figure here, which is not in your book. And what I would like to do here is mention a very important statement, which, if this is going to appear on a national board, will show up in a statement form on the national board. You don't have to memorize this, but in the sickle cell beta globin allele, there's a replacement of a normal A nucleotide by a T, which destroys a restriction site for an endonuclease called MST2. So you know from pathology as well as biochemistry, when the A got mutated to a T, that converted the amino acid glutamate to valine, and as a consequence of that, the beta globin protein is then malformed and doesn't work properly. But now a mutation of the A to a T can be used in genetic testing. Now again, here there's a mutation. Remember earlier we talked about polymorphisms due to mutations. Now here's an example of a mutation which gives rise to a defective palindrome. In this case here, the endonuclease MST2. So what we see in the separate part of this figure here is right here. Let's assume now this is a chromosome, and part of this chromosome is shown here is the beta globin gene. So this is the beta globin gene as part of a very long chromosome. Now what the dark boxes and the open boxes represent is that the beta globin gene has 3 exons and 2 introns. No big deal here. Just all you need to know here is this is the beta globin gene. Now in the vicinity of the beta globin gene, there are 3 palindromes for MST2. And that's going to be illustrated here with the magenta. This line here is a palindrome for MST2. Here's another palindrome, and here's another palindrome. Notice how 2 of these palindromes are within the beta globin gene, and this palindrome shown here on the left is far removed from the beta globin gene. But there are 3 palindromes in the vicinity of the beta globin gene. There are going to be other palindromes in the rest of the chromosome. No big deal here. But within beta globin, near the beta globin, there are 3 palindromes for MST2. Now, according to this statement at the top of this figure, there's a replacement of the normal A by a T, which destroys a restriction site for MST2. Let's assume now this is where that A is that got converted or got mutated to a T. So now this palindrome is no longer here. It got mutated. So now in the sickle allele, where are the palindromes? There's a palindrome here, and there's a palindrome here, but this palindrome here got mutated because the A was converted to a T. Now notice the fragment sizes, the RFLP. Notice the fragment sizes that are being generated with a normal allele for beta globin versus a sickle allele for beta globin. Because there's palindromes here, here, and here. In the normal beta globin, there'll be a fragment size of 1.15 kb in size, right there. There's another fragment being generated of 0.2 kb in size. Now in the sickle allele, this palindrome got destroyed. What is now the size of the fragment? The fragment now is 1.15 plus 0.2, which is 1.35 kb in size, because this palindrome never got cleaved by MST2. So if you take a normal allele or a sickle allele for beta globin, add MST2, we generate these fragments. And then you do a southern blot. These fragments are separated on the basis of size. Now the issue becomes, how are those fragments detected? So we need to have some sort of probe. So now let's bring in a probe. Here's a probe shown here in the red horizontal line. This is a probe that doesn't even see the beta globin fragments. It could be outside the beta globin fragments, but importantly, what this probe is going to see is a fragment size of 1.15 kb in length, and that's indicative of a person who has a normal beta globin gene. Whereas that same probe, if it's added to a sickle fragments, that probe will see a fragment size of 1.3 kb in size, as shown here. This large fragment of 1.3 kb in size is indicative of a fragment coming from a sickle allele for beta globin. Okay, so now the issue becomes, coming back to this pedigree, by RFLP analysis, we can determine what is the outcome of the fetus. Is the fetus normal, homozygous for sickle cell, or heterozygous for sickle cell? So what's done is to take DNA from all 5 of these family members, including amniocentesis of the fetus. So let's assume now DNA is taken from this male here. It's the DNA, the chromosomes are taken. Their MST2 is then added, and then the fragments are then subjected to a southern blot. So again, this is a southern blot. Any fragment size that's detected by this probe, what's the probe? Here's the probe. Any fragment size that's detected by that probe that's 1.15 kb in size is indicative of a normal allele for beta globin. Any fragment that's detected by the probe that's 1.35 kb in size is indicative of a sickle allele for beta globin. So now, let's come back to this male, the father here. So if this father's DNA is taken and then you add MST2, notice there are 2 bands that appear. One band that appears of 1.15 kb in size, saying that at least one of his chromosomes has a normal beta globin gene. And in addition, another fragment is seen of 1.35 kb in size, saying his other chromosome has a sickle beta globin gene. So 2 fragments, 1 normal, 1 sickle. Similarly with the mother here, 1 fragment seen at 1.15 kb, another fragment seen at 1.35 kb, indicating that both the mother and the father are heterozygous for sickle cell. Now what about this son here, who has, the pedigree shows that this son is homozygous for sickle cell anemia, and the RFLP analysis will verify that. Notice when DNA is taken from this son, there'll be 2 overlapping bands of 1.35 kb in size. No band appears of 1.15 kb in size, saying that this person is SS. This person has 2 sickle genes for beta globin. Now this daughter, the pedigree shows is normal. It's also possible this child could be heterozygous for sickle cell, because on a national board they do not have to show you which people are heterozygous. But in this case here, the RFLP will confirm that this daughter is indeed heterozygous, is homozygous normal for beta globin. Why? Because when this daughter's DNA is taken, a single band that appears at the size of 1.15 kb. No band appears of at 1.35 kb, meaning this person does not have any sickle beta globin allele. This person only has normal beta globin genes. Now lastly, with the fetus, what is the outcome of the fetus? Notice that one band appears at 1.15 kb in length. Another band appears at 1.35 kb in length. The outcome, therefore, is what? That this person is a carrier for sickle cell anemia. So the pedigree says this person's a 50% probability of being AS, but the power of the RFLP says this person is. You can change the verb from could be heterozygous to is heterozygous for sickle cell. The power of the RFLP analysis. Okay, so now on this figure here is the northern blot. Now what we've done earlier was the use of southern blots, because we're always analyzing DNA fragments using a DNA probe, but now this is a northern blot. And this is going to be a northern blot of the FMR1 gene. Now first of all, before we talk about a northern blot, I want to ask a very simple question. What does a northern blot measure? And what does a northern blot measure? It measures not just messenger RNA, it measures messenger RNA levels. The amounts, the relative amounts of RNA can be analyzed, not just RNA, but RNA levels. That's going to be very important when we go through this particular figure here. So what this figure represents is a northern blot of the FMR1 gene. Now I'm going to be very precise in asking you a question here, because it might be on a national board, the way they word the question. Suppose they say to you on a national board that this is a northern blot of skeletal muscle, brain, liver, testes, lungs, pancreas, and the heart. Now what I plan, and then you see this particular northern blot. Now I'm going to ask you 5 questions based on this northern blot, and we'll start off with some very simple questions and progressively get harder in the questions. So the first question, if you look at that northern blot, the first question is the gene expressed in the brain? And the answer is yes, the gene is expressed in the brain based on this northern blot. Now how do you know that? Because if you go to the brain lane, here's the brain lane, notice there is a band that appears. So what this is saying is the brain has a messenger RNA for FMR1, which can be detected using a DNA probe. So therefore, the gene is expressed in the brain. So that's a very simple question, a very simple answer. Next question. Is the gene being expressed in the liver? And the answer is no. The liver has many messenger RNAs, it expresses many messenger RNAs, but not one messenger RNA is the messenger RNA for FMR1. Even though it has many messenger RNAs, but none for FMR1. So the answer is no, the gene is not being expressed in the liver. Next question. Why the darker signal for the brain compared to the lung? So you go over here to the brain, notice how this brain band is much thicker than the band in the lung. Why is this one darker compared to this one in the lung? And the answer is that the higher expression level in the brain. So again, notice over here, what does a northern blot measure? It measures messenger RNA levels. So what this is saying is the brain has much more messenger RNA for FMR1 compared to the lungs. Now the next question, when you look at this question, you might give a quick answer, but you need to think about it very carefully here. And the question is the gene present in the liver? So and the answer to this is, because you might say, well, if the gene is present in the brain, it's got to be present in the liver. But the answer is yes, if these organs or tissues are coming from the same individual. Notice at the beginning when I was trying to be precise in stating that here we have tissue from muscle, brain, liver, and so on. If I didn't say that these came from the same individual, then you cannot say that the gene is present in the liver. After all, how do you know the liver didn't come from some sort of elephant? So if these came from the same individual, then the answer is indeed yes, but otherwise you need to do a southern blot. What I'm driving at is, on a national board, read the question carefully. If they showed you a northern blot and they say these bands, these messenger RNAs are coming from the same individual, then the answer is yes, the gene is present in the liver. Even though it's not being expressed, it is present in the liver if it's from the same individual. Now the last question is, it's a hard question, why the smaller size in the heart? So here is the heart. Notice this band is much smaller in size compared to the bands in the brain, testes, and lungs. Why is this band smaller in size? And the answer is kind of a hard answer, but you have to think back to other things in the previous chapters in biochemistry. And the answer to this is, it could be due to possible alternative splicing. Remember how we talked about before what alternative splicing is about, how you can take out certain introns and splice together various exons, and the mature messenger RNAs will have different sizes. So what this is saying is, during the splicing of the FMR1 RNA, the primary transcript, here we have mature messenger RNAs, which are different in size compared to the messenger RNA that's being expressed in the heart. Okay, so I will take a break and then we'll proceed with this chapter. In this figure here, we'll be talking about the basis of the polymerase chain reaction, a very powerful technique that's used in science and medicine. And what the polymerase chain reaction is all about, sometimes you might see it, is called the PCR reaction. It's the same thing. But PCR simply means, polymerase chain reaction simply means amplifying DNA. You can take a little bit of DNA and make mega quantities of it. It's a very easy technique to do in the laboratory, and so that's the basis of what we want to talk about here, the basis behind the polymerase chain reaction. So if you have DNA in a test tube and you want to amplify just a certain portion of the DNA, what are you going to need? What kind of tools do you need to amplify the DNA? What you need to amplify the DNA is you need some sort of DNA polymerase, because after all, you're going to be making more DNA from a DNA template. And then what you also need is excess primers, because remember, way back in biochemistry in the earlier chapters, we had talked about all DNA polymerases require a primer. In your body, the primer is an RNA primer that's needed by a DNA polymerase, but in a test tube, a DNA polymerase could use an RNA primer, or it can use a DNA primer. Now, in the PCR reactions, it's DNA primers that are used. And where do the DNA primers come from? These are primers that you commercially purchase, or you can make in the laboratory with your own hands. But these primers are going to be they're going to be complementary and anti-parallel to the sequence of DNA to which you want to amplify. It's the primers that they're going to direct to show what DNA is going to be amplified. And then lastly, because we're going to be making DNA in the PCR reaction, we need excess amounts of all 4 deoxynucleotide triphosphates, because DNA is going to be made. DNA polymerases need deoxy ATP, deoxy CTP, deoxy GTP, and deoxy TTP in excess amounts. So to amplify DNA, you need a DNA polymerase, excess amounts of primers, and excess amounts of deoxynucleotide triphosphates. So in the top of this figure here, let's assume this is a part of a chromosome among 46 chromosomes. Now, in part of this chromosome, let's assume now this part right here, shown here in the shaded gray, this is a portion of the DNA of a chromosome that we want to amplify for whatever reason. So we want to make tremendous quantities of that portion of the DNA, make double-stranded DNA at the very end, just of that size of DNA. So again, this is a portion of one chromosome among 46 chromosomes, and we want to amplify it, make mega quantities just of that double-stranded DNA. So how is this done? So what's done is take somebody's DNA, put it in a test tube, get rid of all the histones with detergents or something like that, or salt. But now you have naked DNA in this test tube. What happens is we add the primers. Again, the primers are going to look at the DNA, all 46 chromosomes, and look for DNA to which it will base pair with. And so what's in this test tube, we take the test tube and heat the test tube. And when you heat the test tube, the 2 strands of double-stranded DNA will come apart. So now all 46 chromosomes, the strands of the chromosomes now will come apart. And then when the temperature now goes down, as you cool the DNA, the strands, the double-stranded DNAs want to come back together to base pair. But now, because there's excess amounts of primers around, because there's lots of primers around, the primers preferentially will find the DNA first before the 2 strands come back together. So to illustrate this, here's double-stranded DNA. This is a portion of the DNA that we want to amplify. If you heat the DNA, the 2 strands come apart, and then during the cooling process, the primers are present. The primers preferentially will base pair. So outlined here in the red represents 1 primer, which will base pair with strand 1. And then on strand 2, there's another primer that base pairs with this portion of strand 2 of that particular chromosome. Now, regarding strand 1, what's going to happen when DNA polymerase is added, as well as all excess deoxynucleotide triphosphates, all 4 deoxynucleotide triphosphates? What's going to happen is the DNA polymerase will take this primer shown here in the red, and it's going to read the DNA template, in this case here, strand 1. It's going to read it in the 3' to 5' direction. So it's going to take that primer and go all the way to the end of that portion of the DNA. So now, what does the newly synthesized DNA look like from strand 1? That's shown here, outlined here in the red. This is the newly synthesized DNA coming from strand 1. This portion here represents the original primer. This lightly shaded gray portion here represents the sequence of DNA that we want to amplify. And this open rectangle here, the rest of the DNA, just represents DNA that we're not interested in. Now, this is what happens with strand 1. What about strand 2? Here's the primer that was added. It base paired with strand 2 at this position because it's complementary and anti-parallel to it. The DNA polymerase will then use that primer and read the DNA template 3' to 5'. It's going to take that primer and go all the way to the end of that DNA. What does that newly synthesized DNA look like from strand 2? Now it's shown here in the red. So again, here is the primer, the original primer. The lightly shaded gray represents this portion of DNA from strand 2 that got made into newly synthesized DNA. And this part right here, the open rectangle, represents DNA that we're not interested in. Now this is at the end of cycle 1, which takes about a half an hour to 45 minutes. You heat, strands come apart, you cool, the primers base pair preferentially because they're present in excess, and then you add DNA polymerase as well as deoxynucleotide triphosphates, and now it makes these newly synthesized DNA that's shown here in the red. So at the end of cycle 1, we have the original strand 1, newly synthesized DNA coming from strand 1. The original strand 2, and newly synthesized DNA coming from strand 2 at the end of cycle 1. Now in the same test tube, no more additions. In the same test tube, all we need to do again is the heat. When you heat, all these strands then come apart during the cooling process because the primers are present in excess. Now the primers preferentially base pair. Why do they preferentially base pair? They're present in excess. So now here are the primers, now base paired with the respective DNAs. And then the DNA polymerase. Now the DNA polymerase will take this primer, go all the way to the end of the DNA, and now that's going to be shown right here. This is that newly synthesized DNA that we're really not interested in yet. But now the DNA polymerase will come and take this primer and take that primer, go all the way to the end of the DNA, and now notice we're starting to get DNA of the exact size that we're interested in. It's a single-stranded DNA at this point, the exact size that we're interested in coming from the original strand 1. Now over here on the right, the DNA polymerase will take this primer, go all the way to the end, and there it is right there. Now notice again, it's a DNA of the exact size that we're interested in. This is a single-stranded DNA at this point here of the exact size we're interested in. Now this is at the end of cycle 2, yet another 45 minutes or so. Now this is at the end of cycle 2. Now we can continue to heat and cool, repeated heating and cooling every 45 minutes. There's going to be amplification of the DNA in an exponential manner. So at the end of about 20 cycles, basically that just takes overnight, at the end of 20 cycles of heating and cooling, we end up with DNA of the exact size that we're interested in. This is double-stranded DNA coming from original strand 1. This is double-stranded DNA coming from strand 2, from the original strand 2. This is at the end of cycle 3, but after about 20 cycles or so, we're now ending up with multitude of copies of DNA of the exact size we're interested in. And theoretically, after about 20 to 30 cycles, millions of copies of that exact size of double-stranded DNA that we're interested in. So again, way back at the beginning, we might have had 46 chromosomes. We're only interested in amplifying a piece of it, but now we have amplified tremendous numbers of that double-stranded DNA that we're interested in. So this is the basis of the polymerase chain reaction, PCR. Now, what I want to do now is discuss several applications of the PCR reaction. And before we do that, what I want to do here is give you a sample question, which is in your book. and talk about this in reference to the importance of the primers. It's the primers, as I mentioned earlier, it's the primers that will find the DNA that you want to amplify in 46 chromosomes. So here is a question that comes from your book. I'll give you a chance, I'll give you about 10 or 15 seconds to read this question. If you don't get to the answer, we'll give you the answer, how we get to the answer, in just a few minutes. So I'll give you about 10 to 15 seconds to read the question. Okay, so this is a patient who has Huntington's disease. And it says, which of the following, there's a question here, which of the following sets of primers would be used to amplify the CAG repeat in the brackets? And the brackets is shown right here. What kind of set of primers would be used to amplify this? Again, you got 46 chromosomes, and one of the chromosomes you have the CAG repeat. How do you find it? In other words, what kind of primers are being used? So this is a single strand of DNA. On the left is the 5' end. On the right is the 3' end of this DNA. Now, how do you know this is on the left is the 5' end and on the right is the 3' end? Because you must assume it. On the national board, if they do not tell you which end is the 5' end or which end is the 3' end, if the numbers are not given to you, you must assume on the left is always the 5' end. So this is the 5' end because nothing's told to you. This must be the 5' end on the left. So this is strand 1. Now on strand 2, because of in double-stranded DNA, what would the strand 2 look like? What strand 2 would look like is now shown for you here. This is strand 2, which is complementary and anti-parallel to strand 1. So now, what do we want to amplify here? What we want to amplify is shown in the brackets is this portion right here. We want to amplify the CAG repeat. So this is the CAG in strand 1, but in strand 2, it's also CTG from the 5' end to the 3' end. So it's CTG. So it's this part that we want to amplify. So how are we going to amplify just this part among 46 chromosomes? So we need to add primers that are near this part of the DNA that we want to amplify. Now, one primer that's going to be used that flanks this part of the double-stranded DNA, one part is shown here. This is one primer that's going to be used. Notice how this primer is complementary and anti-parallel to strand 2, this part of strand 2. This is 5', G, A, G, T, C, C, and so on, which is complementary to this part of strand 2. Now, in strand 1, what is the primer that's going to be used? That primer is now illustrated here. It's the 5' CGG, CGG, and so on to the 3' end. So this primer is complementary and anti-parallel to this portion of strand 1. So remember, in DNA synthesis, in replication of DNA synthesis always occurs 5' to the 3' end. So what's going to happen is the DNA polymerase will look at this primer here, that's base paired to strand 2, look at this primer, and then go from, in this diagram, go from left to right because it's going to make DNA 5' to the 3' end. So this part is going to be amplified. Another DNA polymerase will look at the primer that's base paired to strand 1. Take this primer, go from right to left in this diagram, again amplifying this part of the DNA. So as a result, we'll end up with tremendous numbers of the CAG repeat. So the answer to this question is D. Why is that? Because so the two primers are going to be used. One primer is this one shown here. Notice how this sequence is identical to this part right here. This is the 5' GAG and so on, the 5' GAG and so on. Now this part right here, this is the little tricky part. You're going to look at this primer and look at that primer and say, hey, these are not the same sequences. But remember, this is the 5' end to the 3' end. 5' C, G, G, C, G, G, and so on. Now down here, if nothing is given to you, you must assume this is the 5' end. Let's draw it in there. This is the 5' end. 5', CGG, CGG, which is now identical to this primer here, 5' CGG, CGG. You must assume this is for any sequences of DNA. If the numbers are not given to you, you must assume on the left side is the 5' end. So that's why the answer is D. Okay, so this is a sample question of the, and also illustrates the importance of the primers for amplifying a particular part of DNA. So now let's talk about the applications of PCR reactions, not in the sample question here, but for a technique, in this case here, for paternity testing. Okay, so on this figure here is an application of the PCR reaction in paternity testing. Now another technique here, it's in paternity testing, it's the use of PCR in genetic fingerprinting. So in this example here is some child, and here's the mother of the child, and so we want to test some male to see if this male is the father of this child. So what's done is genetic fingerprinting is a very good technique to determine if some male is a father of a child, and this is going to be using VNTRs. Now earlier in this chapter we talked about VNTRs, which is another name for it is microsatellites, also called mini satellites, depending on how big the sequence of the DNA is. But in this example here, we're going to talk about a VNTR ACAT. So in the mother, let's assume now that in the mother, in one of the chromosomes, this VNTR ACAT is located somewhere in a chromosome, and it's in tandem 7 times. So it's a 7-mer. ACAT is amplified 7 times in a row, in tandem. That's in one of the chromosomes. In their other homologous matching chromosome, let's assume that same VNTR ACAT is amplified 10 times. So this would be a 10-mer. Now some tested male, so let's assume some father of a child, in the same chromosome that ACAT is also a 7-mer, and in the father's matching chromosome, homologous chromosome, that same ACAT is amplified 15 times in a row. So it's a 15-mer. So again, this mother has a child, and we want to see if this male is the father of that child. So what's done is to take the mother's chromosomes and then amplify a particular VNTR. In this example here, we're going to amplify the ACAT VNTR. So how do you do this again? You got to add primers that flank the ACAT. If you add primers in the test tube with the mother's chromosomes, the primers will base pair with on the flanking sides of the ACAT, and by PCR reaction, the ACAT will then be amplified. There'll be a 7-mer coming from one of the chromosomes, a 10-mer coming from the other chromosome in the mother. The same primers are then added to the DNA from the chromosomes from the tested male. So here are the primers on the flanking sides of the ACAT VNTR, and so in one of the chromosomes from the tested male, there will be a 7-mer, and in the other chromosomes there'll be a 15-mer because the PCR primers are added, and this part will be amplified. So then after the PCR reactions from a test tube containing the mother's DNA as well as a tested male's DNA, the amplified DNA is then subjected to a Southern blot, and so this is what we have here in the Southern blot, where DNA is now being separated on the basis of size. So if they're separated on the basis of size, the 15-mer, the position of the 15-mer will be indicated right here. The position of the 10-mer is going to be indicated right here, and here's the position of the 7-mer. So here is the DNA that got amplified from the child, again using the same primers, taking a little bit of DNA, the chromosomes from the child, add the same primers, amplify the DNA, and here's the PCR amplified DNA from the child, and here's the amplified DNA from the mother, and here's the tested male. Now the way you do these paternity testing Southern blots is look at the child and say which bands of the child came from the mother. So you look at the two bands in the child, which band had to come from the mother? It's this middle band here. It's the telomer had to come from the mother. Then you look at the other band in the child and say, did the tested male have that band? And the answer in this example, the answer is yes. That during inheritance, this band here, this chromosome, original chromosome over here, had to come from the tested male and given to the child. So what is the conclusion from this type of southern blot? The conclusion is this tested male could be the father of this child. Now we'll do this one step further. And this is another diagram in your book. Let's assume it's the same VNTR, ACAT. So again, the way you do these things, you start with the child and say which band in the child came from the mother. So you look at the child band and you say which band came from the mother. It's this band down here. And then you look at the other band in the child and say, did the tested male have that band? And the answer is no. So what's the conclusion? The conclusion is this tested male could not be the father of this child. So now let's assume you get the situation shown here on the left, where a band did come from the mother, another band did come from the tested male. The conclusion there is the male could be the father of that child. So how do you get rid of the verb could be to is the father of that child? The way that's done is 4 to 8 different VNTRs would have to be tested for a definitive identification. So this is one VNTR, ACAT, but 4 to 8 different sequences of DNA would have to be tested. And if all cases that there's always matching, the child did have a VNTR that was inherited from the tested male, at that point there you can get rid of the verb could be to the word is the father of that child. So this is used in paternity testing. This can also be used for this genetic fingerprinting technique. This can also be used for forensic medicine and things like that. So now another technique, another procedure in which PCR reactions are used is for HIV testing. So if someone comes into the clinic and they want to be tested for HIV, the first technique that's generally used is ELISA assays. The reason why ELISA is used, as you know from immunology, it's used because it's very cheap and very quick. But the problem with ELISA assays is that it can be false positive. And the reason for this is cross-reactivity. Antibodies in the blood might cross-react with proteins from other sources. So you can't have the problem is being false positive. And then what's done to see if someone has HIV, as you know from immunology, another powerful technique is the Western blot. And so Western blots can be used, ELISA assays can be used, but the best technique to determine if someone is harboring the HIV virus, the best technique by far is the PCR reactions. And when is PCR generally used? And the most common reason why PCR reactions is used is when a newborn whose mother is HIV positive. Now why is that? Again, this harkens back to immunology, is that if a mother has HIV, the HIV antibodies are passed from the mother to the newborn in the milk. So, so if a mother has HIV, then the newborn also will have the HIV antibodies, even though the newborn might not have the virus, but certainly would have the antibodies because they're being passed on from the mother. Another reason why PCR reactions can be used for testing purposes, it can be used for early testing after known exposure to HIV positive blood, after some sort of finger prick or something like that where someone's known to have HIV, then you'd have to be tested. So what is done there in PCR reactions for HIV testing? What's done there, it's like so if someone has 46 chromosomes and HIV has now integrated into the chromosomes, it's like trying to find a needle in a haystack. How do you find that particular HIV genome in 46 chromosomes? Again, trying to find a needle in a haystack. So what's done there is do PCR reactions. So again, now here, it's the primers that's going to be directed against a particular gene of HIV. These primers might be directed against the GAG gene or the envelope gene or the Pol gene. It doesn't have to be against the entire HIV genome, just but one of the genes of the HIV. So in this case here, in this example, let's assume we have primers against the GAG gene. So in the laboratory, we make primers that are complementary to the GAG gene. And so then we take somebody's DNA, put it in a test tube, and then add the primers along with DNA polymerase as well as all 4 deoxynucleotide triphosphates. And now we're going to PCR amplify the GAG gene. So PCR amplify by using HIV-specific primers. And then after PCR amplification, we then take the amplified DNA and then do a southern blot, and then you probe with what? Again, in this case here, because we're interested in the GAG gene, we have probes that are complementary and anti-parallel to the GAG gene that got amplified by PCR. And then by southern blot, we can then see a band that's now detected. So in this case here, it's not the entire HIV, but it could be just one of the genes in the HIV genome. So a very powerful technique, the PCR reactions for determining if someone has HIV. Now, the last figure in Chapter 7 is the quantifying of viral load in HIV infections using something known as RT-PCR. RT stands for reverse transcriptase PCR. So if someone has HIV, as you know, HIV is an RNA virus, so the RNA can be in the blood. And so what's done is to take somebody's blood, and then if you want to determine how much, what's the viral load, how much virus is present. So you take somebody's blood, and then you add reverse transcriptase, which will take the RNA and make complementary DNA, because reverse transcriptase takes RNA and makes DNA from that. And then what happens is you take the complementary DNA and PCR amplify the cDNA, and now we would have PCR amplified cDNA. So in a test tube, we take somebody's RNA, add reverse transcriptase, and make cDNA, then amplify that cDNA by PCR. Now the question is, what is the viral load? So in order to determine that, we need some sort of standard curve. And so what's done is to take the PCR amplified cDNA, let's assume we have this amount, whatever that number is, let's assume it's 1 million copies, and then we go to the standard curve and then straight down. This will tell us this is a number of DNAs that were obtained from the RNA originally per unit volume. In this case here would be per mil. So this is quantitating the number of viruses in the bloodstream by amplification using PCR. If there's no RNA virus in the bloodstream, you add reverse transcriptase, there'll be no cDNA, and so nothing can be amplified by PCR, and then there'll be a zero RNA, zero DNA will be shown up by looking at the standard curve. But if someone has lots of RNA in the bloodstream, you add reverse transcriptase, then PCR amplify. The more RNA in the blood, then by looking at the standard curve, there'll be more copies of the DNA per unit volume. So we have now finished with Chapter 7.